All right, so today's lecture, transgenic plants and animals. The questions we're going to talk about are, what is a transgenic organism? And how is a transgenic organism created? And as you'll see, what we're talking about today has implications both for research, in terms of being able to manipulate organisms so that you can study them, and also in terms of a lot of important things that have to do with society, for example, our food supply and our health. So the kinds of things we're talking about today are of increasing importance, and I want to give you an idea of scientifically what's going on, because there's a lot of misinformation and misunderstanding about transgenic organisms that I hope we can clear up and then make more clear what the societal implications are. So let's start with a really simple definition of what a transgenic organism is. So this is one definition of a transgenic organism. It's an organism that contains DNA from another species and that can pass on this DNA to its progeny. So sometimes a transgenic organism contains DNA from the same species, but that doesn't really matter. This definition will do fine for us. And the important part of it is not only that it has foreign DNA, but that it can pass the DNA on to its progeny. And usually what this means is that you have this piece of foreign DNA, and somehow that piece of foreign DNA gets integrated into the chromosome of the organism that is the host for this DNA. And then it behaves from that point on as if it's part of that chromosome.
So this is like the germline theory?
Yes, yes. But it's not limited to humans, obviously. This could be any organism that we want to talk about. Yeah, in the sense that it's therapy, right? It's a medical treatment, yeah. Okay, so you've seen examples of transgenic organisms already in this class. In fact, you've dealt with them in the lab, so you've actually made a transgenic organism in a sense. Because you've taken a piece of plasmid DNA that was in a test tube, and you mixed it with some bacterial cells, and those bacterial cells took up that DNA, and then those bacterial cells passed on that DNA to their progeny cells. Okay, so that's a transgenic organism, and you've seen this picture before. And basically, you can have any piece of DNA you want inside a plasmid vector. Okay, and the bacteria will take up that DNA and keep replicating it and behave as if it's part of their normal DNA. And so as you know, the first step in the process is inserting your piece of DNA into the plasmid. And then the second part of it is that once that's inside a bacterial cell, the cells multiply and then keep making more and more copies of that piece of DNA you want. You've seen this before. And we can sort of summarize the laboratory process by saying you have this recombinant plasmid, a plasmid with a piece of foreign DNA in it. And then what do? You do what you did in lab. So you take E. coli cells and you mix them with the plasmid in solution. And then with the right salts and a little heat pulse, which you did in lab, the bacteria will take up that plasmid. And then you can culture those bacteria on a plate of agar containing nutrients, right? And also containing an antibiotic, because the plasmid confers antibiotic resistance, and so that's how you know that you have that piece of DNA there. Okay, so this is something I talked about in lecture. It's something you did in lab. And the result is that you can make as many of cells as you want like this that contain that plasmid, and therefore make many, many copies of that piece of DNA. And if a bacterial cell doesn't have the plasmid, it'll die because it's not resistant to this antibiotic ampicillin. Right? Okay? So the way I want you to think about that in terms of today's lecture is that there's really two steps in this process. There's a delivery step. Somehow you have to get the DNA into the organism that you're trying to make transgenic. Okay? And in the case of the plasmid and the bacteria, you basically mix them together with the right salt, and then you give them a heat pulse, and that's how you deliver the plasmid inside the bacterial cells. So there's a delivery phase, and then there's a selection phase where the delivery is never 100% efficient, and the plasmids won't stay in the cells unless you force them to keep the plasmid. So there has to be a selection phase too, and in this case, the way the selection is done is using this antibiotic drug. Okay? And that's how you know you always have that plasmid. So those two steps, delivery and selection, are what's crucial to making any organism transgenic. Okay? And if you think about it, most organisms, that's more difficult to do than bacteria. Okay? And this is going to be a little bit crude, but it's a biology class, so I'm allowed to do this, right? So think about how you deliver foreign DNA if you were trying to make a transgenic human, okay? You have to get it into the gametes, right? It doesn't matter if you put DNA into somebody's skin cells. That's not going to get passed on to the next generation, right? In order to make a transgenic human, you have to deliver the DNA to the right place. Okay? And then once you deliver the DNA, you can't just plate humans down on a petri dish, okay, with an antibiotic to make sure that they're transgenic. Okay? So when we're dealing with a complicated multicellular organism like a human being or like a fruit fly or a nematode worm or a plant, you have to figure out some way to do the delivery and some way to do the selection, or it's not going to work. Okay? So what I want to show you first is how it's done in flies, because it's just sort of a standard way. But it also shows you how, with a little bit of cleverness and a little bit of understanding of the organism that you're studying, you can do this. And so I would venture that for any particular organism that you cared about, you or someone else could figure out a way to make it transgenic. Okay? It just takes a little bit of cleverness. So here's how you make a transgenic fly. So first I want to show you what a fly embryo looks like. And you've seen pictures like this before, but not so big. Okay? So this is a fly embryo right after a female has laid it. Okay? So very, very, very young embryo. This is the head end. This is the tail end. Okay? There's actually a lot of yolk in this egg. That's why it's sort of dark in the middle. That's what a fly embryo looks like. Okay? And so what's unusual about flies, and this is what I said, where it takes some knowledge of the organism and some cleverness to do this right. What's unusual about flies is that the cells of the embryo that will actually become the larva and then the adult are going to form only around the outside of this football-shaped thing. Okay? All of the inside, as I said, is yolk. And what's really unusual is that those cells don't actually form by making cell membranes in between them as they divide. What happens first is all the nuclei divide, and then the cell membranes form. And that's unusual. Usually, as nuclei divide, cells divide, and you get new cell membranes. But in the first couple of hours of a fly's life, all of the nuclear divisions happen, and then the cell membranes get made. Okay? And what that means is that you can insert DNA in solution into this embryo, and you don't have to poke it right into a particular cell. You can just get it into the embryo, and then just by diffusion, it'll end up in a bunch of cells. Okay, usually a cell membrane would be a barrier to that diffusion. So the way you make the transgenic flies then is you first collect a lot of these, a lot of very, very young embryos, okay? And that's actually pretty easy to do. You get yourself a milk bottle-sized container, and all of these spots here are flies, right? So this probably has a couple hundred flies in it. And it's about, this big, right? Like a little drink bottle, okay? And you put on the end of it a plate that has sort of like a bacterial plate, right? It's made of agar, but it has grape juice. Basically grape juice and agar, and that flies love grape juice, and so they'll go right to the grape juice and they'll lay their eggs there, okay? And the only thing they like better than grape juice is yeast, okay? So if you put a little dab of yeast on grape juice plate, the flies will go crazy. And so they'll lay lots and lots of eggs on these plates, and then you can collect the plates, and you have lots of embryos. So then what you do is you go to your fancy microscope, because obviously these fly embryos are really, really small, right? And you have to insert DNA into each embryo, okay? And so I'm going to show you the parts of this setup in a second, but basically you have a microscope. You know how to use a microscope, right? And what's right here actually is an arm that contains a very fine glass needle on the end. And this arm, you see, is hooked up to this really complicated contraption. And what that allows you to do is basically, like a video game, control that arm really precisely. Okay, because if you tried to, take a big needle like a syringe and jab it into a fly embryo, you'd probably miss, and if you didn't, you'd probably destroy the embryo. So you need this really fine control to be able to inject the fly embryo, and that's what this contraption is. And you're basically looking through the microscope at the embryos and lining this thing up so that the DNA gets inserted into the embryo. Okay, so if you take a close-up on basically what's sitting here, right, where your microscope slide would be, is you have a microscope slide on which you've lined up a bunch of fly embryos. Okay, each of these horizontal lines, it looks like a zipper almost. Each of those horizontal lines is an embryo. Okay, and here's this fine glass needle, and you can see this green right here. That's just food coloring, so you can see that there's a liquid solution in the needle. Okay, and the needle's basically going to each one of these embryos in turn and stabbing the end of the embryo, as you'll see in a second, and inserting some of that solution, a really tiny volume of that solution into each embryo. Okay, and it's actually sort of like a video game. I spent a lot of my days in graduate school doing this, right? You just sort of line up your embryos and you go pow, 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 right? And you hope that you're getting the DNA in there and that you're not killing the embryos so that these things get a chance to grow up and you actually get some transgenic flies. Okay, so this is what the delivery phase looks like in really high close-up. So this is the tail end of the embryo because it's at the tail end that the cells that will form the gametes are going to form. Okay, so back here, there are a bunch of nuclei, and some of those nuclei will eventually belong to cells that are forming the germline. Okay, that is the cells that will form gametes that can be passed on to the next generation. And so here's the needle coming, right, towards the tail end, and you stab the embryo, and it actually recoils a bit, right, because you're stabbing this thing into the tail end of it. And then it's hard to see here, but if you've printed it out, you'll see that there's basically a green splotch right here, because the solution in the needle was green, okay, and then it gets inside, and the only reason we use the green food coloring is so we can actually see what's going on. And so you get this big spot of green liquid inside the embryo, and then you draw the needle back out, okay? And if you do this right, you don't cause too much damage. The embryo, it sort of seals itself back up, but you've inserted DNA into it, okay? And so that's part of the problem, right? That's the delivery part of the problem, okay? But then the question becomes, well, okay, so now you have a solution of DNA inside the organism, but how does that actually get into the chromosomes? Because as I said, for a standard multicellular organism, in order for that to get passed on from generation to generation, it actually has to, that DNA has to take up residence within the chromosomes so that it behaves as if it's part of the chromosome. So that's the next challenge. You can get DNA into the organism, but now that DNA has to get its way into the chromosomes, okay? And the way that's standardly used to do this is using what are called transposable elements, or sometimes colloquially they're called jumping genes, okay? And you've seen one example of this already that I'll show you in a second. But basically the idea of a transposable element, which we sometimes abbreviate TE, is that they're a piece of DNA that can move from place to place in a chromosome. Okay, so let's say there's a TE sitting on this chromosome over here, and this is another chromosome. Basically what a TE will do is it'll make a copy of itself and jump that new copy into the chromosome somewhere else. Okay, so you already know an example of a TE, okay, that's taking up 10% of your own genome. Okay, anyone remember the name of that TE? Yeah, it's an ALU element, ALU element, okay? So 10% of your genome is that one particular TE that basically just keeps jumping around. And then there are lots of other TEs in your genome that jump the same way. And the same is true in flies, the same is true in worms. TEs are pretty much ubiquitous, okay? And the idea, right, is that you can harness these TEs to your own purposes, okay? They are jumping around people's genomes, you know, sort of as we speak, right? But you can also use that ability to jump to your own advantage. And I showed you one example of this, but I didn't really explain it that much at the time. Much earlier in the course, when we were talking about this minimal organism, this Mycoplasma genitalium, which is a very small genome, very small number of genes, only a few hundred genes. And we could ask the question, well, how many of those genes are actually essential for that organism to live? Right? And the way I said you tested that is you disrupt each gene one at a time and ask if the organism can still live, right? If it can still grow this particular bacterium, right? And I had this graphic here where I say, okay, you disrupt it with a big yellow triangle, okay? But what that is actually is a transposable element, okay? So the way that experiment was done is they got this one transposable element to jump a lot, right? And then sometimes it would jump into this gene, sometimes it would jump into that gene, sometimes it would jump into that gene, and so on and so on. And then later on, you can figure out which jumps are compatible with those cells still being alive. Okay, so that's one example of harnessing a transposable element to do a particular type of experiment. You can also harness them to carry DNA. Okay, so the idea is that if you take a transposable element, which I had as a big blue rectangle, okay, and you take out part of that transposable element and you put in your favorite gene there, then the transposable element will jump around with your DNA, okay? And the reason it does that is because for most transposable elements, the only sequences that actually matter for it to be able to jump are at the very ends, okay? And basically everything else in the middle just gets carried along for the ride. And so what that allows you to do is put whatever DNA you want in the middle, and then that can jump, okay? And so if you want a particular transgene in a fly, then you take a fly transposable element, you put your favorite gene, which we call a transgene because it's now being used to make a transgenic organism, okay? And then this transposable element will just jump into the fly's chromosomes, okay? So the steps in the process are a little bit more complicated than just doing a bacterial transformation, but not much, okay? So you start out with a plasmid, and the new thing about this plasmid is it has these ends of a transposable element in it, only the ends, okay? And then you add your favorite DNA fragment that you want to get into the fly in between those two ends. So now what you have is this transposable element that contains your gene and is capable of jumping that whole thing into a chromosome. So that's the delivery part of it. What's the next part that we have to worry about? Selection, okay? So in general, we don't use antibiotic resistance as a way of selecting transgenic flies, okay? They're not bacteria. We don't use ampicillin to select when we have a transgenic fly. So we need some other way to figure out which flies actually have the transgene, okay? Because the other thing to remember, too, is that in this case, the whole plasmid doesn't enter the chromosome. Only this part of it enters the chromosome. So we need something on here that's useful for selection so we know when we have a transgenic fly, okay? And so, again, this is a place where some cleverness comes into play. And so early on, when people were trying to do this with flies, they realized there were a lot of mutant flies that were known that had messed up eye color, okay? The very first mutant that was isolated at Columbia about 100 years ago was a fly that had white eyes instead of red eyes, okay? And about almost 30 years ago, the gene that was mutated in that fly was discovered, okay? It's a gene called white, and it encodes a pigment transporter that makes sure the red pigment gets to the right place, okay? And so what you can imagine doing is taking flies like this as the ones that you inject and having as part of your transgene this white gene, okay, that is required to make the fly's eyes red, okay? So what we do is we take our embryos from this strain of flies and we inject them with DNA that looks like this, okay? So we have the blue ends, those are the transposable element ends, okay? And we have whatever piece of DNA we really care about, which I'm calling the transgene here. And then we have a marker here that allows us to tell when the transgene is there. Because in most cases, the transgene is not something that you can automatically select by itself. You want to study that in flies, but it might not actually show its existence in any particular way that's easy to detect. So you need something else to know that the transgene is there. Same way you need ampicillin resistance to know that a plasmid is there. Okay? And so one of the things you can use is the normal gene for red eyes. Okay? So the normal gene that's mutated in these flies, if you take the wild type normal version of that gene and put it back in the flies, you expect to get red eyes. And that's what you get. Okay? So the other half of my time in graduate school, after I did the video game of injecting the flies, was looking through hundreds and hundreds and hundreds of white-eyed flies to find that one that was red. Okay? And then you can actually start your experiment, because then you have your transgene inside the fly and you're studying whatever you're studying. Does that make sense? Okay. Same kind of tricks are used to make transgenic plants. So, but again, it has to do with the biology of the organism. You have to know the biology of the organism in order to do this in a rational way. Okay? So what's used in a lot of plants is something called Agrobacterium. Agrobacterium, it's a bacterium, and it's harmful to plants in general. So if a tree like this one is infected with Agrobacterium, you get these things called galls that are basically big tumors on the plant, right? And what the Agrobacterium does is it infects the plant by injecting its DNA into the plant, okay? And part of the DNA that it injects carries genes, right, that encode enzymes that synthesize plant hormones, okay? And so if you have the wrong plant hormones being produced in the plant, then what you get is this uncontrolled growth of these cells, and you get this big mess of a gall, okay? And so people had studied Agrobacterium. They'd figure out that they had figured out that this is the way Agrobacterium works, that it injects its DNA into the plant as its way of causing pathogenesis in the plant. And so they figured, well, if the bacterium is injecting DNA into the plant, then maybe we don't have to, right? We can use the bacterium to get the DNA that we want into the plant. We can trick it to taking our DNA in instead of its DNA in, okay? And so the DNA that the Agrobacterium is inserting into the plants is actually also a transposable element. It's normally called tDNA, so it has these ends on it that are what gets recognized and inserted into a new chromosome, okay? And normally, Agrobacterium tDNA carries a bunch of virulence genes here, the genes that cause havoc with the hormone production and so on, okay? But what you can do is you can take all of those virulence genes out and just put your genes, okay? So you can put your transgene that you want to study, and you can put some kind of selectable marker here too. And for plants, actually, the most common selectable marker that's used is actually an antibiotic called kanamycin that works on bacteria, it works on yeast, it works on plants. It's just a useful way of selecting transgenic plants. Okay, so I want to show you a little cartoon animation that these people at Purdue came up with to show you how it actually works in plants, okay? And again, it's kind of clever the way they figured out how to do this. So, this will work. All right, so that's one way that you can transform a plant. The other way, one of the groups mentioned, oh, sorry. Yeah.
Sorry, so when is the tDNA inserted? It's already in the agrobacterium, it's going to be inoculated?
Yeah, so basically it's sort of two steps of transgenics. First, you make your transgenic agrobacterium that's carrying the genes you want, and then you use the agrobacterium to deliver that to the host. So, right, so one of the groups actually mentioned this in their presentation last week, but another way that's used in plants and also in some animals to do delivery of a transgene is actually what's called a gene gun. Okay, and so that's a gene gun right there. And you can see this poor plant that's about to get shot, right? And the way this works, again, is really clever. So basically what you do is you take a bullet and you coat it with a solution of particles of either gold or tungsten, some kind of heavy metal, that are coated with DNA in solution. Okay, so you take these particles of gold or tungsten and you wash them in a DNA solution so that those particles basically have that solution attached to them. And then you take your bullet and you coat that with the particles. And then you shoot your gun, okay? And in the first iteration of this, it wasn't a gun that looks like this, right? It was a gun that was a rifle, okay, an actual rifle. And so you take your gun with this DNA-soaked bullet, and you fire it at a piece of metal that has a hole in it, like a donut, okay? And basically that donut hole is too small for the bullet to go through. So what happens is the bullet hits that hole, and it stops, but everything that was sticking to it keeps going, right? So these gold or tungsten particles that were attached to the bullet keep flying through the end, and that's basically what's happening here. So there's a place here where a projectile stops, but whatever was attached to the projectile keeps going at very high speed, and those particles of gold or tungsten penetrate cells, they get into the cells, and then the DNA can basically dissolve off of those particles and is now inside the cell. And so then that DNA is capable of entering the chromosomes of that plant. So the nice thing about this is that you can basically try it on any organism you want, right? So it's done for various types of plants that have never been transformed before, like papayas. Some plants can't be infected with Agrobacterium. They're actually resistant to Agrobacterium, so you can't use Agrobacterium to transform them, but you can always try and fire DNA into them. And so it's a fairly general, useful tool. Sometimes it's used for nematode worms and other organisms too. Okay, so, and you can hear Robert Horsch talking about the gene gun on that clip if you want to. So the next question for us is, so we know how to make transgenic organisms now. And as I said, you know, whatever your favorite organism is, there's bound to be some way that you can make it transgenic. And then the question becomes, well, what do you use it for? What do you make transgenic and why? And this is where you start running up against, well, what are societal priorities? What are the potential benefits and drawbacks of using transgenic organism? And so the first transgenic organism I want to show you is kind of a laughable example, but it sort of gets to this point right away, which is what's the point of going through all this trouble to make transgenic organisms? And so these are what are called glowfish. They're transgenic zebrafish with fluorescent pigments in them. Okay, so under a normal aquarium, which usually has some kind of UV or black light, fluorescent light on it, these glow these bright colors. Okay, and so if you want, you can get a transgenic pet, okay? And your transgenic pet will be nice and colorful. Unless you live in California or Canada or most of Europe, because those places have rules against transgenic fish, okay? And so the original purpose of these fish, according to the company that makes them, was not for them to be these cute little pets. But actually, they claim that the original purpose they wanted to get this technology going was that so that fish like this could actually be environmental sensors, right? And so that they wouldn't always glow, but maybe they would only glow if there was mercury in the water, or only glow if there was, you know, if the pH was too low, or there was raw sewage, or whatever. And so, in principle, one could take a bunch of these zebrafish, let them out into some environment and see if they glow. And if they glow, you don't go swimming, right? If they glow, you try and clean up that runoff or whatever. That hasn't happened yet. I find it kind of far-fetched that would ever happen, because you could also just go and take your pH meter. You can go and take your, you know, mercury sensing kit and do the same thing instead of releasing transgenic organisms into the wild, right? But at least in terms of the company's PR, that's what they claim to be doing. And you might ask, well, why can't you buy them in California? Why can't you buy them in Canada? Why can't you buy them in Europe? And the reason, basically, is that those places have laws on the books against transgenic organisms somehow being released into the wild. Okay, so for example, transgenic fish. And so you can see how even like a trivial example like this, like having pets that glow, becomes wrapped up in sort of what are society's interests. You know, a consumer might have an interest in having a nice, cute, glowing fish, but one of the reasons California doesn't allow these in is because there hasn't been an environmental impact statement done on these fish. And so the company would have to pay probably hundreds of thousands of dollars to do the research to show that these are actually safe for the environment, they're not going to cause any problems, and they just haven't bothered to do that. They're willing to forfeit that market. Is the what? Yeah, that's plastic, I think. Maybe it all comes as a kit, right? You get the nice glowing seahorse too. Okay, so on to some more sort of serious examples. Very recently, okay, so last year, the first transgenic primates were made. Okay, so these are marmosets. They're not that different from us. Okay, again, if you can make a transgenic marmoset, you can make a transgenic human. Okay, it's not that different, right? And the selectable marker that they used is actually the same kind of fluorescent protein that was used in the fish. Okay, it's green fluorescent protein. Okay, and so this is a baby marmoset. This green right here is actually a latex glove. So this is the researcher holding the marmoset with a glove. Okay, so it's tiny, right? It's just fitting in someone's hand. Okay, and then the real kicker is when you look at the marmoset's hand. Okay, this is a normal marmoset hand, and this is this marmoset's hand. It's glowing green. Okay, and that's how they know that this marmoset is transgenic, right? And if this marmoset grows up and has kids, it'll have green. Those baby marmosets of the next generation will also have green hands because it's a true transgenic organism. Okay. So yeah. So how many years is it going to take before we see a green human hand? I don't know. And what I want you to realize is that it's not a scientific issue at this point, right? If this marmoset lab was given the go-ahead to create a transgenic human with green hands, and everyone in that lab was okay with doing that, they could do it. I'm sure they'd have you a transgenic human with green hands in a year or so. Okay, so at this point, it's really not a scientific question of whether you can do that. It's a societal question of whether you can do that, and currently, you can't do that. By far, the main use of transgenic organisms in society with a huge, huge economic and social impact is transgenic plants. And these are transgenic plants mostly that are sort of staple food crops, but also things like cotton as well. And so transgenic plants have been made for quite a while and used for various things. And there are a bunch of things you might consider using a transgenic plant for. Okay, so this is just a fairly comprehensive list of the kinds of things you might want to do, might want to use a transgenic plant for. So let's say you had a plant that was a food crop, okay, but it's always ravaged by insects in the field. Okay, so one thing you might want to do is make it resistant to its most common insect pests. Okay. You might want to make it resistant to bacterial and fungal diseases. I've talked earlier in the class about making it resistant to herbicides so that you can basically take a crop dusting plane and spray your whole field with that herbicides, and only your crop will grow because the crop is resistant to this broad spectrum herbicide. You might want to increase the nutritional value, and we'll talk about an example of this in a second. The vitamin content, the carbohydrate, protein, lipid balance of the food, you might want to adjust that somehow. We've talked about sort of increasing yield, so the idea that you would make the plants produce food in a more efficient way. We've talked a little bit about increased stress resistance when we talked about rice and the idea that some strains are more resistant to salt and others are not, and when you have these marginal environments that people are using to grow food on, then it becomes more and more important that the plants can actually tolerate the various stresses they're exposed to. I'll show you an example of production of pharmaceuticals. That is using plants to make drugs so that you could actually take drugs by just eating something as opposed to getting injected with it. Or potentially growing up those plants and purifying the drugs from them and then injecting them. improved marketability for various reasons. Okay, so this is the first transgenic organism that was marketed for food. It was the flavor saver tomato, almost 20 years ago. And the idea was that part of the problem with tomatoes was that you would try and ship them across the country and they'd all spoil. Okay, and so if you could make a tomato that would not ripen until you told it to, then that might be an advantage. And so these tomatoes were made, and they basically were made so that there would be a trigger that would allow them to ripen. And in principle that works, but the tomatoes basically tasted horrible. They were made from a variety of tomatoes that had other undesirable properties, and so they didn't last very long. They're basically a failure economically. And then the last thing on this list here is sterility. So you might ask, well, why would you ever want to engineer a plant to be sterile? And the reason is so that you can make money. So if you think about it, if you're a seed company and you sell somebody some seeds for a plant, well, the plant's going to make its own seeds at the end of the season, right? And then that person could get those seeds and plant the next year, and then get the seeds from that and plant the next year. And that's how people have been farming for thousands of years. If you're a seed company, that might not sound like a great business model to you because you're basically giving someone something that lasts forever. And so if you want people to buy seeds every year, you either have to improve your seeds every year so that you're making a better crop all the time, or you make it so the seeds grow into plants that can't make seeds. And then the farmer is dependent on you, so you have to buy those seeds every year. So here's one of the common modifications of plants. You'll see in an upcoming lecture sort of how the percentages play out in terms of what crops are actually transgenic, how much of that crop area is actually transgenic, and what purposes the transgenes serve. This is one of the common ones. It's a way to make plants resistant to insects, okay, by having them produce a toxin internally, okay? So this is a bacterium called Bacillus thuringiensis, BT for short, okay? And actually, if you ask organic farmers, they'll tell you about BT, okay? Because it's been one of the sort of organic ways of dealing with pests for a long time. You can actually get the bacteria, you make a sort of mixture of it with water, and you put it in a spray bottle and you spray it on your plants. And then the bacteria will be there, and then insects that would have eaten the plants will eat the bacteria and get sick and die. Okay? So that's considered an organic treatment because you're taking one just sort of natural organism that's in the environment anyway, and you're just applying it to a different part. You're not adding any chemicals or fertilizers or anything like that. It's a way of managing insects. But what scientists realized was, well, there's one particular gene in the bacterium that encodes a toxin, and that toxin is what makes the insect sick. And so they figured out a way to get that toxin gene into plants like corn, okay, using the kind of techniques we talked about, using Agrobacterium transformation. So you take the toxin gene from BT, you have some marker gene that you use for selection. You do your Agrobacterium transformation, okay, and if a plant cell does not take up this DNA, it'll die because you're doing some kind of antibiotic selection. If it does take up the DNA, it'll live. You add your plant hormones so that you can get roots and shoots and plant it in the ground, and you get a corn plant out of it. And so that corn plant now has a corn genome that has this bacterial toxin gene in it. That toxin gene is activated, and it makes the toxin. And so if there are caterpillars that are crawling on the corn plant and trying to eat it, which is a common source of damage to corn plants, they'll get sick and die, okay? Dead caterpillars, right? So this works quite well. There are debates about whether this is a good idea, because if you think about it, if you had the bacteria on the plant making the toxin, that's fine. When you harvest the corn and wash it off, you wash off all the bacteria, okay? But here what you have is corn where basically every cell in that plant is making the toxin, and you can't wash it off. It's inside the plant cells. Okay, so when you eat this corn, you're eating that toxin. Now, probably doesn't do you any harm. There have been debates about whether it does harm to other organisms. So, for example, one of the things people worry about is, well, some caterpillars we actually like. Okay, people are really attached to monarch butterflies, for example, which migrate long distances. They're pretty, and people like watching them. They don't want them to die on cornfields. And there's some evidence that that the widespread use of Bt plants, pollen gets lots of places. Monarch butterflies don't eat corn plants, okay, but they eat other plants that might be nearby. They might be subject to this toxin, and it might cause them problems. And then you have a species that then gets threatened by the widespread use of this toxin. And as I said, there are arguments on both sides about whether it's a problem or not. As I said, you could use plants in principle to make pharmaceutical products. So this is an example of using plants to make vaccines. Okay, so you all probably know the principle of a vaccine, right? So you take some organism that would cause you harm, and you take usually some dead version of it, and you inject it into your arm. Your body reacts to it. It mounts an immune response against that organism that can't really cause you any harm because it's dead anyway. But the fact that you've mounted the immune response to it, then if you actually get exposed to the real living organism that can cause disease, you won't get a disease because you'll have this memory effect in your immune system, and you'll attack that right away, and you won't get sick. Okay, so that's the principle of basically every vaccine. And the idea is, well, what if you could make a tomato or a banana plant produce fruit that contains a vaccine? Okay, and bananas are seen as a really interesting case where this might be useful because kids like bananas. Okay, it's easy to get them to eat bananas as opposed to get a needle jabbed in their arm. And bananas grow in parts of the world where vaccine delivery is not particularly great. And so some people see it as a way in sort of tropical third world countries of delivering vaccines perhaps more efficiently than they have been in the past. And so people are pursuing this. And again, the way you do it should not be a surprise to you. You take, say, a banana leaf. So this example, which is from an article, I think, same article you'll read, is with a potato plant, okay, but it could be a banana plant, right? You take the leaf, you have this cut edge, you put on your Agrobacterium suspension to infect that, insert the tDNA, select it on an antibiotic medium, and the tDNA contains genes from whatever you're trying to vaccinate against. So instead of vaccinating by, killing a bunch of streptococcus bacteria and sticking them in your arm, you can figure out which proteins are the ones your body actually mounts an immune response against, and just get the bananas or the potatoes or whatever to actually make those proteins. Okay, and again, just making the proteins is not going to make you sick because you don't have that full organism that can actually cause an infection. So you get your potatoes or your bananas, and now they're basically a normal edible potato or banana, but in principle at least, it's a vaccine also. It'll protect you from some disease. So these are not in widespread use. This is something that's being researched. Another example, which is actually my favorite example of a really, really good use of transgenics. Okay, so there's a spectrum, right? You can take people who are opposed to genetically modified transgenic organisms no matter what. Okay, there are people like that. And there are people who, if there's an economic reason to do it, then do it. Okay, we're a capitalist society, go for it, right? And so there's a spectrum of how much caution and how much investment and all these things in transgenic organisms there should be. Okay, and so I to be honest, right, to be sort of full disclosure, I'm sort of towards the spectrum of, you have to have a really, really, really good reason to make a transgenic organism for me to be in favor of it. And the reason can't be to make you richer. Okay, that's sort of my personal philosophy. You're welcome to disagree. Okay, but here's an example where even me, right, even this sort of leftist professor who can't stand people making money off of transgenic organisms, even me, I say this is the kind of use you'd want to put it to. Okay, and so let me describe this case to you. These are Hawaiian papaya fields from 1994. Okay, and what was happening in the early 90s was this virus called papaya ring spot virus was wiping out the papayas in Hawaii. And what you can see here actually is a bunch of stumps of trees. These were papaya trees that were cut down in an attempt to stem the spread of this virus. Okay, an attempt, these were infected trees. They were cut down, burned, trying to get rid of the virus. It didn't work. So these other trees here are all infected, and you may be able to tell that these are not what papaya trees should look like. They're really stringy looking. Their leaves are really in bad shape. You can see in this one, for example, it's basically dead. This virus is really virulent. The papayas do not do well when they're infected with this virus. Okay, and so this was a huge problem for papaya farmers in Hawaii at the time, and a group of scientists figured out a way to make papayas resistant to this virus. Now, if you think about the sort of standard way you might go about trying to make resistant papayas is you would try and do breeding, right? You try and select plants that were capable of surviving the viral attack. But if you look at this field, there are no survivors, basically. You can't, I mean, you can try and grow as many papaya trees as you want, but there weren't any papaya tree varieties that were really good at withstanding this virus, okay? What the scientists managed to do, though, was through transgenics to make the papayas resistant to the papaya ring spot virus, okay? And so these are two papaya plants that are identical, except this one has a transgene that makes it resistant to the virus, and this one doesn't, okay? And both plants are infected with the virus, so basically they were both sprayed with the virus. This one is in deep, deep trouble, and this one is perfectly fine, okay? It's basically immune to the virus. So how did they do that? They did it basically the same way that you would make one of you immune to a virus. They figured out a protein that was on the coat of the virus. So this is an electron microscope picture of the virus. It looks like a long string, okay? And basically, there are proteins that are lining the outside of it that are called coat proteins. They coat the virus. And all they had to do was take one of these coat protein genes and get it into papaya plants, okay? So you use your gene gun. You get your coat protein gene DNA on those tungsten or gold beads. You insert them into the leaf cells. They get taken up by the leaf cells. You have some kind of selection, antibiotic selection, for example, to know that they got in. And then you have a papaya plant that is a normal papaya plant, except it's making a protein that's normally found on the virus. And it's not really clear why that makes the papaya plants immune, okay? If this was a human being instead of a papaya plant, it would be very clear why. Because we have an immune system that recognizes foreign proteins, and then our immune cells attack those any cells that have those proteins on them. That's how a vaccine works on you. Plants don't have the same kind of immune system as we do, so it's not really clear why this works, but it does. Okay, and it works amazingly well. So here is a test field in Hawaii, where all of the papaya trees, so it's one big square, but all the papaya trees on the outside are not transgenic ones, and all of these are transgenic. Okay? So the virus basically wreaks havoc on the non-transgenic variety. So if you look at sort of the borderline between transgenic and non-transgenic, it's really clear. These plants are basically dying, and these plants are perfectly fine. And it wasn't just Hawaii, okay? So Hawaii was one of the first places where it was clearly becoming a major problem. but this was a papaya orchard in Taiwan that was affected by the same virus. This is papaya trees in Thailand that were affected by the same virus. Here are the transgenic ones right next to the not transgenic ones. You can see the gigantic papayas that are growing on these trees. These trees are perfectly fine. Okay, so to me, this is a great example of doing something with transgenics that you couldn't do otherwise. There wasn't really an option, right? You're basically saving an industry that was sort of a unique industry. Growing these papaya plants was sort of the livelihood of a small but important group of farmers in places that don't have a lot of other options in some cases for making money. Okay, and so to me, this is a great example of doing something for good. Another example that's sort of along these same lines, but a lot more controversial for good reason, is trying to do something about vitamin A deficiency in humans. Okay, so this is a map of basically places in the world where vitamin A deficiency is a big problem. Okay, and as I've said before, vitamin A deficiency is the leading cause of childhood blindness. And so the places in red and orange are the places where it's the biggest problem. Okay, you can see that as you might expect, in developed nations, it's not a problem at all because people eat varied diets. They don't have vitamin deficiencies in general. And so the question is, well, how do you help the part of the world where this is a serious problem? One idea that's been put forward is that you engineer the staple crop that people are eating anyway to produce vitamin A. Okay, and this is something called golden rice. Rice does not normally produce vitamin A in the endosperm. So the endosperm is basically the seed kernel. That's the rice. So this is normal white rice. This is golden rice. The reason it's golden is because it's producing beta-carotene, which is the precursor to vitamin A. Okay, and beta-carotene, as the name suggests, carrot is yellowish orange. Okay, and so the idea is, well, you can make this golden rice, and then people would eat it and they'd get more vitamin A than they did if they were eating that. And so the scientific problem then becomes, well, how do you make this rice? Okay, and to do that, you have to figure out, well, why is this rice not producing vitamin A? Because lots of parts of plants produce vitamin A. And the answer is that in the seed kernel, there are two enzymes that are missing. And if those two enzymes were there, you would get vitamin A produced. You'd get beta-carotene produced. Okay? And so you have this biochemical pathway, and you've seen examples of biochemical pathways a few times before in the course. There are these chemicals. It doesn't matter what they are. They're just abbreviations here. But there's this one called GGPP that's a precursor for a lot of compounds within plants. Okay? And you can see that by this biosynthetic pathway, eventually you get carotenoids like beta-carotene. So this is the pathway that goes to vitamin A. And it turns out that in the kernels of rice, two of the enzymes in the pathway are not produced. Okay? And so you can't get from here to here. And so you don't get vitamin A out of rice. But all of these other things work perfectly fine. So GGPP can get converted to gibberellins, for example, which are the growth hormones, as you know. And so the trick then to make golden rice is basically to get these two enzymes to be produced in the rice kernel. Okay? And so the names of these two enzymes are PSY and CRT1, and basically they catalyze these steps in the pathway from GGDP all the way down to a bunch of things, including beta-carotene. You don't need to know any of these details, but the point is if you can get these two enzymes produced in rice, you'll get beta-carotene. And so this has been done successfully. The trick, right, is to get it expressed in the right place. So rice will produce these enzymes, but just not in the kernel. So how do you think they get these enzymes produced in the rice kernel? How would you do it if you had to do it? Well, so you definitely have to get a transgene in there, okay? But what would the properties of that transgene be? How would you design that transgene so that it would be active in the rice kernel?
Well, it's like the endosperms of the gamete, right? So you'd have to make sure that that section of DNA within the gametes somehow, like in the slide where you inserted it into that section of the embryo.
So the kernel itself is past the gamete stage, right? It's sort of the embryo, right? So there's two questions. There's the question of how you get DNA into the plant, and we've already discussed how you do that. You have to have some kind of delivery system. You need Agrobacterium or a gene gun or something. But then you have to get these enzymes activated in the right place. So how would you think about doing that? What kind of information would you need that might help you to do that? Yeah, so close, right? So basically what you need is some sequence that's a promoter of a gene that you know where that gene is expressed in the kernel, okay? Similar to what we talked about with like the stickleback fish, where the different promoters cause the activity of the genes to happen in different tissues of the fish. There are different promoters that cause different genes to be activated in different tissues of a plant. Okay? And so if you had a gene that has a promoter that's active in the kernel, in the actual rice, then you can hook up that promoter to the coding sequences for these two enzymes, right? And then you have a promoter that's active in the right place, and then the enzyme that you want to be activated there. Okay? So that's basically what was done. And that gives you, instead of rice that look like this, you get rice that look like this. And then version 2.0 of golden rice looks like this because they figured out how to get even more production of beta-carotene, so it's even more yellowish orange. Okay, actually 20 times more provitamin A in the new stuff than the old stuff. Okay, so this all sounds well and good. It's not in production yet because there are field tests going on now. The idea is once you make one rice plant that has these transgenes, you want to get it into the varieties that grow well in different parts of the world. So you think about sort of introducing those genes into the elite cultivars that are used in various parts of the world. So for example, there's a Japonica strain. This golden rice 2 strain is a Japonica strain. But if you wanted to get indica rice, you know, a long grain rice, then you have to somehow cross this gene now into an indica strain to get that going, right? There are field trials in progress now in various places. And this, again, is a very controversial thing, right? And it's controversial, I think, for good reason. It's complicated, too, because this is, in a sense, a philanthropic effort, right? The scientists who are in charge of doing this, who came up with the idea for this, have, I think, excellent intentions. The effort is now supported by the Gates Foundation. It's a real philanthropic effort. It's not, you know, a corporate thing that's being sort of forced on farmers. But there are a bunch of issues, right? So one issue is, did anyone consult the sort of populations who this is supposed to benefit to ask if this is the way they want to solve this problem? Okay. The answer to that case, as you probably know, is usually no. Vitamin A pills are actually quite cheap. And if people could afford it, they'd probably eat a varied diet. Okay, so maybe the solution to this problem is not to prevent people from having vitamin A deficiency. The solution to this problem is to give them economic opportunity, because then you wouldn't have vitamin A deficiency either. So in a way, it's seen as sort of curing a symptom as opposed to curing a disease, right? There are people who worry about the safety of transgenic crops. And so this is something, you know, that I think the scientific jury is still out on, but as a scientist, as a geneticist who works with transgenic organisms, at least in the laboratory, I'm not afraid of them. You know, I don't think eating them is going to cause much harm in general, unless you're messing with sort of nutritional pathways in a way that you don't really understand. But just the fact that an organism is transgenic, I don't think makes it dangerous to eat. And in fact, you probably are not aware that, you know, much of your diet is actually transgenic right now. You're probably just not aware of it because you don't live in Europe, where everything has to be labeled. So, you know, the safety issues I think are there, and there's always the burden on the producer to prove that what they're selling is safe. But it's not what worries me the most. The things that worry me the most are sort of the economic precedent that it sets, because the idea is that you have this sort of top-down solution to everything. You have widespread use of things. Even though this is a philanthropic effort, there are other efforts that are sort of coming right behind that don't have that sort of same kind of philanthropic goal. And we'll talk about those, you know, I'm going sort of from really good examples of what you might want to use for transgenics to the really bad examples. So next lecture is like the real disaster uses of transgenic. And this is sort of somewhere on the borderline, because I think it has the same kind of mentality as some of the really bad uses, even though it's coming from good intentions. The other thing that really worries me as a biologist is ecological problems, okay? So anytime you have a widespread planting of any particular crop, you have a potential disaster waiting to happen. Because, and we usually call that monoculture, so the idea that you're planting one plant variety over large regions of your arable land. And the reason is that, you know, in any one year, some new pest might arise, some new disease organism might arise, some really bad weather conditions might happen. And if you're planting the same thing everywhere, you're at risk for any one of those things really destroying an entire crop, okay? So in that sense, sort of variety, having a bunch of different plants growing is a really good strategy. It's basically hedging against all of those potential disasters that could happen. And one of the scariest statistics I heard recently is that as of not too long ago, there were 5,000 different varieties of rice that were grown in India, okay? Across the world, many more varieties. As of today, it's more like 50. Okay, so we're definitely, and this is true of all major crop species. Okay, we're definitely going to this state where larger and larger tracts of land are used for fewer and fewer crop varieties, and so, and there are examples of crop failures that are really dramatic. Okay, so you know the most famous historical example is the potato famine in Ireland, right? But if you think about the way plants are grown now, the potential for something much bigger than that to happen is actually really high. And it's getting higher because you have the same plant grown over large areas of the globe. And that, to me, is a scary prospect that could be prevented by smart, you know, policies that promote a variety of plants being grown at any one particular time. Okay, and as I said, next time we're going to talk about some really awful things people do with transgenic plants.